Okay, for people joining this live stream, we are talking about, as the title of the notebook says, biological sequence processing in Wolfram Language, and we have both internal folk and some external experts here. So let's start going through this. All right, what is this change listing? What does this mean?
Just a summary of the changes as a result of this work.
So where should we start? Should we start on this, or should we start on the, we talked about this last time, right? But
that's right. There's
new or different?
There's new and added, yes.
So how should we go through this? Should we start at the top here, or should we go through?
I think start at the top.
Okay.
I mean, the first one can be very quick, just to get an impression about the scope.
Okay. There's a lot of biofunctions, but then our market is humans who are biological, so I suppose it's not surprising that we have a lot of bio-related things here. That is a very questionable title. I mean, a very questionable name, I think. Okay, so we're gonna go through. We're going to go through all of these. Is that correct?
That's right. They're all represented here. Yeah.
Okay. Okay, and so these are various underlying. So we've talked about a bunch of this stuff.
Yes. But you wanted one longer meeting where we one time coherently go through again from the beginning.
Good.
Because we didn't make any final decisions, naming wise.
Okay. Well, let's take a look at this. This is interesting. Okay. So we have genes as entities. We have proteins as entities. What about like animals as entities?
For animals as entities, those can be entered into find biosequence features to obtain features of the sequences that have been discovered from their genome.
Okay, but so in this diagram, they feed into things here, presumably.
Right. So the animals as entities would feed into the find biosequence features, which is currently an arc from biosequence DNA down to biosequence position, segment, and mutation at the bottom there.
Okay, so biosequence. Okay, so biosequence. I mean, that's not really that's not the right term. Amino acid. It's really that is protein. DNA is to protein. Base pair is to amino acid, in a sense. So that's not an amino acid. That's a biosequence of amino acids. That's a biosequence of. I mean, the overarching thing is DNA there and it's protein there. Correct?
Maybe not quite, because not any three amino acids don't make yet a protein.
Okay, so what would those be called?
Peptides, maybe?
That sounds like a good name. That's what people. Yeah, that's the right name, isn't it? I mean, Okay, all right. Okay, so then that's interesting that there's a concept of so biosequence position is the analog of like a geoposition or something.
That's right. Yeah.
And it has the analog of sort of a datum, which is the which build of the human genome it is, and all kinds of stuff like that.
That's right. If you click on the plus there, you'll see an assembly.
Okay, and so biosequence segment is. I see. By the way, we're hearing clicking, typing. Biosequence mutation. So that's basically a SNP. But could it be anything other than a SNP? Like if it's some kind of big insertion of a bunch of stuff, or a repeat or something?
Yeah, that's right. And not only that, SNPs aren't precise. They can be many different variants. And so this gives more information by telling which particular variant is observed.
Okay. This looks pretty cool to me. Yeah, am I confused, or is this wrong relative usage here? I mean, that's a DNA sequence, but this is, I don't know, isn't the analogous thing here peptide? I don't know.
I really like the peptide suggestion. I think we should take that forward.
Okay. I don't quite know why these icons have to be so monochromatic, but that's a very detailed issue. I don't know why this is what is a mixed sequence?
Yeah, so this is kind of one sequence kind of concatenated after another, joined in some, in this case, unspecified way.
When does that happen? What's that?
That happens in our lab all the time. We make conjugates of like RNA, DNA, or DNA and peptide and that kind of thing. Or you'll get it with glycosylated sequences as well. Although I guess that would have to do with branching. Can this do that?
It can. We need to talk more about how to specify the different joins and conventions for that, but that's kind of where things need to be resolved now.
Got it.
So what is unspecified? What does that mean?
That's when a user hasn't entered any type associated with the biosequence. It's useful for catching if people forget to say, just forget to type in DNA and want the result returned, or just genuinely have some type of biosequence that we don't yet support. That's some new thing that they found in their lab, and it's our way to handle that flexibly.
So wait a minute, but once and we'll get to this, I'm sure, once you've got a biosequence in there, for DNA, it's, letters representing base pairs or or uncertain combinations of those and so on. I mean, there's a definite coding of how we represent the different things that actually appear in DNA, correct?
That's right.
But so for unspecified, how does that, I don't understand what that is. It's just like, I'm calling it W, and I'm going to tell you later what that means, or what?
That's right. It's for things like Hajimochi DNA, where before they had named it Hajimochi DNA, if they wanted to do some work on it in their lab, they would still be able to represent it as something unspecified.
Okay.
It comes up a lot in bioorganic chemistry. There's things like PNA and peptoids, things that usually share sequence but have very different backbone chemistry.
I see. Okay, so here's a naive chemistry question for you guys. You're talking about conjugating things. Okay, this is a super naive question, but you got a DNA strand. Can you glue a piece of RNA onto the end of it?
Yeah, it's called a conjugate. You absolutely can. The linkage chemistry can be different. That's what he was saying. We have to figure out how to sort that out in mixed sequence, like how they are connected specifically. But there's lots of different reactions that you can conduct off the end of one strand to concatenate it to another.
Does biology use those things?
It does for glycosylated proteins, for instance. It will stick carbohydrates off the end.
Oh yeah, that I knew. Yeah, right. But okay, fair enough. I see. I just hadn't heard of it with DNA.
Often do that as far as I know, I don't think there's peptides hanging off of DNA or RNA that way.
Yeah, and glycosylation.
Chemists will do it in the lab all the time, but it doesn't show up in biology, I don't think.
Sorry, was Trevor going to say something? What?
Yeah, I'm familiar with glycosylation, but that being just kind of marking different peptides along the sequence rather than actually filling in gaps in the sequence. So for working with things, not that it's not useful for other things, but I would never have this mixed sequence object to work with.
Fair enough. Okay, let's dig in then. What is this broader functional picture?
These are some of the extensions that we've thought about for where to take this work after having covered what we've implemented.
Okay, let's dig in. Biosequence. Creating a sequence. Okay, so this first argument, can that be an entity?
Yeah, yes, it can. Yep.
Okay. And so this funky versions of DNA and so on, those are entities, is that correct?
So right now we've supplied 3 base kinds, and we've also supplied a way for users to extend the listing that's supported, as you'll see subsequently.
Okay, but isn't it the case that there are okay, I'm asking what kind of a thing, so there's an entity which I can put in as the first argument of biosequence. What is the type of that entity?
Biosequence type.
Okay. But isn't it my understanding that, like, critters that live in random hydrothermal vents or some other weird sulfur, I don't know what they are, but that they have different, I don't know exactly what I'm talking about, but I mean that they're different.
They do have different mappings between their DNA and then their peptide sequence. Their translation is different, and we do support different translation table entities for that purpose.
I see, but what about between eukaryotes and prokaryotes? Obviously, there's a difference in the.
Yeah, so the DNA and the RNA are typically the same, except for in the transfer RNA can really differ drastically. And that's one reason why we've gone more with sort of extended alphabets for transfer RNA.
So now this thing here, where there's a multi-letter piece in, what does that mean? Does that mean T or A?
Or that means just T concatenated with A, so it's all concatenated together.
So in other words, this thing, each letter, whatever is represented here is represented by an individual letter.
That's right.
And so I could as well represent the sequence as just quotes A, G, T, T, et cetera, as one glob. Is that correct?
That's right. You could just string join it, and it would be equivalent.
What's the canonical form?
String.
So it's joined. So this is merely a convenience that I can put it in a list, but it ultimately ends up as a concatenated string.
That's right.
And we believe that the world of biology and synthetic biology and so on is stable enough that, even if we have to use wild Unicode characters to represent these things, that we're still going to be able to fit them in character strings.
Yeah, that's the presupposition of a lot of great existing work that we should be pretty safe as far as that goes.
Anybody?
The classical thing, the classical thing it's fine for RNA and DNA. The classical thing with amino acids is usually to use the 3 letters rather than the 1 letters that I've seen, but you can make it work either way, I think.
Well, let's talk about that for a second. I mean, so the issue is, okay, the point that I was making is future-proofing this against the kind of, in synthetic biology right now, people are still, very much piggybacking on existing, biochemistry. But I'm just asking a future question here. Can one imagine some sort of variant of DNA with completely different base pairs and so on?
Oh yeah, that exists for sure.
Okay, but so how do people letter those?
They usually give them names like X and Y and stuff like that, and they try to avoid. There are also, unfortunately, certain degenerate names. Like there are letters that respond to that are meant to be like any pyrimidine or any purine.
Sure.
So you have to avoid what the existing.
Okay, so let me make a comment then. So to me, the biosequence type needs to define its alphabet, so to speak. Am I making sense?
Or so, I guess how is that different than each type defining the alphabet that what it's over?
No, that's fine. That's fine. I'm saying that the biosequence type, one of the properties, and maybe you already have this, one of the properties of the biosequence type. So when it's Martian bacteria or something, they will end up with, if it's a Martian bacteria biosequence type, then the letters, when we'll take them still to be letters, will have in one of the properties of Martian bacteria biosequence type is the mapping from those letters to entities that represent what that sequence actually is.
Oh, absolutely, yes.
So in other words, there should be a property that is an association that goes from letters to entities, correct?
Yeah, biosequence type, I just added recently so that each term goes to a chemical. There's an example of that below, and that also can be extended.
Okay, but I'm suggesting that there's specifically a property of that biosequence type which says, T goes to tyrosine or whatever it is.
Yep.
Okay, that's already there. Okay, fine. Well, in that case, I'm unworried about this scheme, because worst case is somebody has some weird synthetic something or other. It's a different biosequence type with a different alphabet.
Yep.
Okay.
Yep.
Let's talk about in the peptide sequence here. Let's just talk about Brian's comment about the 3-letter version versus the 1-letter version.
That may be specific to my experience too. I think there are other people who are used to seeing it the other way. I think it's different school to school.
Okay.
I usually work with 1-letter version.
Okay, but so to me, I could imagine that this entity type here defines which one which scheme you're going to use for that for the payload, so to speak. Were you going to say something different? How does the 3-letter scheme work?
But the 3-letter, you mean the codons or not?
Do you mean the codons, or do you mean something different?
No, I mean there's like met, his, val. There's just 3-letter representations of.
Oh yeah, right. Which have combination uppercase, lowercase letters, correct?
Yeah, usually you do them camel case, and yeah, and they're always 3 letters too, so you can naively break it, I think.
Right. But I mean, so that representation, how do we feel about, if what we ended up with was something that says, PYR, TYR, whatever it is, something like this.
This is great. I could just normalize to the 1-letter form, and then also add a peptide free-form biosequence type that's in free-form natively, and add conversions between the two. And then people could use whichever they'd like, and it would get canonicalized appropriately, and everybody could work in whichever they prefer.
Fair
We
enough.
have a sub-value that returns this type.
Yeah,
That's
why not?
string.
As a string, right? And the string would be smooshed like this. Is that correct, guys? Is that how you usually see it? Is something like that, as a smooshed string?
Yeah, exactly.
Okay. Okay. All right, let's move on here. Default arbitrary number of terms. So that is the unread. That's the sort of the blank.
That's right.
I'm going to ask later about patterns, and I don't know whether this relates to patterns yet.
It does a bit. There's a biosequence match queue which handles these.
I see. So this really is like a blank.
That's right. It's any particular.
Well, I know what it stands for. I mean, I know what the convention is. I'm asking whether, for our purposes, when you do things like, biosequence position or biosequence cases or something, that we are treating this like like in regular expressions, we have a different form of star or whatever. That this is treated like that. Is that correct?
Oh, yeah, for the matching functionality, there is something that will expand it into the expression for matching the fully specified case.
I didn't understand that, but we'll come to that. On our live stream, someone was asking about circular DNA.
That's is an important case that's not explicitly represented here, but
okay. So when you have a circular DNA, what is the framing of the coordinates for it? Is there a standard for that, or is it? I mean, does every circular DNA come with an obvious start point? I'm kind of asking Trevor because I figure he deals with critters with circular.
Yeah, sometimes. Usually you'll start it at the origin of replication is, I think, convention. But obviously it's arbitrary.
But so should we worry for our biosequence representation? Should we worry? Should we have, for example, a different biosequence type called circular RNA or something?
I don't think so.
You might make a higher level construct called like a plasmid or something. When you get closer to genes too, you then want to say like what region is coded for what when people do things like build a plasmid. Mm-hmm. So it might be a higher level thing rather than bottom level like this.
Yeah, but I'm used to, just as a simple example, if you look in GenBank and look at plasmids or bacterial chromosomes, they won't. It will still be a string, and then you'll have a somewhat arbitrary start, but it will often be origin of replication.
Okay. Okay.
There's a couple other edge cases too on the nucleic acids they came up with, like how you would represent methylated positions or end phosphorylation at the end, and abasic sites too is another one that comes up.
Let's talk about methylation, which I sort of vaguely think I understand at least what it is. So you have to kind of annotate the sequence to show where there's methylation. Is that correct?
Yep.
What's the standard for how to do that?
Don't know off the top of my head.
I don't know either. That's kind of a can of worms in that if you think of like human ENCODE, the ENCODE project, in addition to methylation, there's all kinds of marks that you put on functional genomics over like what this is. This a promoter? Is this an enhancer? All of these things, and I'm not actually sure how those are canonically work. I bet it's I know the format. Just 1 second.
One pattern that we have for this is our annotation rules mechanism that we've been introducing in lots of things like audio, for example, where there is there's an audio stream and you don't want to mess with the audio stream, but you want to have annotations that associate themselves with the audio stream. Does that seem?
Yeah, and then it's like you need to know exactly kind of which interval this thing refers to, and it can be complex intervals. Like there will be an enhancer that is not just a contiguous chunk of DNA; it kind of has gaps in it of where it refers to. And
right. So I think in the case of annotation rules, hopefully I'm running a version which will have this in there. Okay. Specify. Okay, so what this is doing is it says annotation rules, and then it's kind of a free form thing where it says where and what. So I think that's the kind of thing we'd probably want here.
Yeah, that seems reasonable.
Yeah, you could use that for all three of those problems: methylation, phosphorylation, and abasic.
Okay. All right, so then the left-hand side of that might be one of your sequence position specifiers, although it's a little weird to put. We're going to come to that because I'm not quite sure whether there's a disembodied position independent of the base sequence that it's talking about. Am I making sense? In other words, when you have one of those bioposition things that you had up here, does that have the sequence inside it, or how does it refer to the sequence?
So this is a feature found from the sequence.
I'm aware.
So this is just the sequence on, say, chromosome 10 in this feature position.
I know what it is. I know what it is. I'm asking biosequence position, right? I'm asking, does this wrapper contain the actual underlying chromosome 10, or how does it refer to chromosome 10? Because what we're proposing here, if we have annotation rules and it says, okay, I've got this gene that lives on chromosome 10, it's at this position, and that's my main event. But then I'm going to annotate that gene with annotation rules that refer to this.
But this would be the segment, not the position. You don't annotate the position, you annotate the sink.
Okay,
the coordinate system.
Fine. Okay, fair enough. But doesn't that segment contain the underlying as well as the specification of where it is? And what I'm worrying about is if you say I'm going to put annotation rules into the underlying object, but the left-hand side of the annotation rules is itself a biosequence segment which contains the object, we're going to get into trouble. Did that make sense?
I think so.
Because here, notice that our Menger sponge, right? Its annotation rules are a part number here that is referring to a piece of the Menger sponge, Menger mesh, whatever it is, but it is not itself saying Menger mesh in there, right?
But there's kind of a convention that this annotation rule refers positionally rather semantically.
Generally, yes. But that left-hand side can be arbitrary based on the type of object that appears, that it's nested inside.
But if it is associated with the object, we always can come up with a convention so that no trouble arises.
Oh, I know, I agree. But I'm saying that the most natural way to do it is whatever is inside this biosequence segment that is the position specifier, we would want a disembodied position specifier for the annotation rules. Whereas we might think we would want biosequence segment arrow whatever, but that's not going to work because the biosequence segment actually has the lump of Menger sponge, so to speak, inside it.
Right.
It's no contradiction to say for the biosequence that was found, there's a methylation on this particular gene that lives inside the gene sequence segment. So as long as the two features are different, that you're not pointing to the same one, it's still okay. You can still say, we're talking about the part of the genome.
I got it. Did you understand what I was saying? The problem that I'm having is
as long as it's
this symbolic object, right, has in it the underlying sequence, okay? But yet we are trying to annotate the underlying sequence. So that isn't going to work. What this would have to have is merely its position information, not its full biosequence segment. And then you'd got a slightly awkward situation where you want to pull out that particular biosequence segment. Anyway, let's just put that on the side. There may be an issue with this, but in the first approximation, handling sort of biology's version of annotation rules with methylation or whatever it is, we should be able to do with annotation rules, I think. Let's see. That's a good point on our live stream from Truth Teller saying circular DNA will be important if you want to find subsequences. That is a good point. How is that, guys who, like Trevor, you were talking about the origin of replication. What happens if you want to find a sequence that spans that? Or is that never going to happen because the thing would never actually be read?
It definitely happens. So there you'd want your pattern matching to kind of append at the edges and just do that wrapping in your pattern matching.
Guys,
Like could be a third argument to something like biosequence match Q.
I don't think that's a good idea. I personally think that you should make the biosequence type be different for circular DNA. I think that will work better because I think that way there's so many weird things that are going to happen with that, like the fact that the pattern matching is different. And it doesn't seem to me like there's a high cost to having lots of biosequence types because I think let me ask you this question. Is there a conversion, like a critter that has circular DNA, it doesn't in its larval phase, I'm making a joke because I know these aren't at that level, but end up with a non-circular DNA? Or does that, in other words, if it's a circular DNA, is it like always a circular DNA, or can it have two forms?
Well, the closest thing will be our mitochondria is circular, whereas our chromosomes are linear.
Certainly in the lab, you can linearize the plasmid. That's a normal thing to do. You cut it.
I see. But you cut it in different places. So then what we would do then when you do the cut is we turn it from, circular DNA to ordinary DNA. Is that right? Or is that a reasonable way to think about it, that one would change the biosequence type from circular to linear?
True, although I guess we'd have to deal with if you have overhangs. Not sure how you do it here, other than we haven't gotten into single or double stranded and what that property looks like.
Okay. Well, so there's a potential issue with this circular. So again, pinning it on the set of issues, a circular DNA biosequence type is what I'm predicting we're going to end up with.
Then I have a question about this subject. So if we have such circular DNA, is it only the fact that within its own loop it is circular, or does then embedding matter? Meaning, like, are different circular DNAs in different topological knots different, or are they all the same?
That's a fascinating biophysical question that I think can be unanswered. They can have things like tension built into them, depending on how they get constructed, and it's
Because if
discussed
this
a lot
is the
about
case, we have infinitely
that
many types.
Yes. I mean, I know
It's a
I've
mess.
seen these pictures of knotted DNA, but is that I mean, there was a big fad for that stuff a number of years ago. Can somebody explain the biology of, does DNA get knotted?
Yes, but I don't think it matters for our purposes. Like, I
Okay,
can't think
fair
of,
enough.
like, it's important for physical packing of the material, and there's all
Mhm.
kinds of isocores and so forth to kind of make this work well. But I don't think it matters for our purposes. But yeah, that's my opinion.
What is an isocore?
So, like, there are these bundles that the DNA, bundles the DNA surrounds, like nucleosomes, and then a higher
Okay.
level isocores, and the DNA is wrapped around these nucleosomes. That is, like, serves to kind of just physically pack things into a small amount of space. And you'll find things like this. We're talking about the markers, where genes that are currently being transcribed, the nucleosomes will have fallen off to allow the transcription machinery to attach. Then they'll kind of come back on when transcription's done. And you can read that out, and you can kind of mark what will be bound by nucleosomes. But I'm not sure the actual kind of knotted
Fair enough.
tangled structure matters.
If we got into that, we skipped a few steps anyway, because there's
Yeah.
secondary structure with single-stranded stuff.
Right.
That's much more interesting to people than the supertension and all that.
Okay, let's keep going here. All right. So these numbers are basically representing patterns of different lengths.
That's right.
Correct? Okay. What is this? I wonder whether that actually makes sense to have the single, that's the single, single amino acid, which is otherwise going to be called peptide. I don't know whether that makes sense. Why do you think that that's a good idea to have the one argument form?
It generalized very well for the generating random instances, but I don't know if it has any real biological utility.
I see. Random instances is interesting. Yeah, okay. All right, fair enough. Okay, biosequence convert. That is very weird. That is very weird to me. The word unspecified being there. I mean, I don't even know should that be automatic or something, or what should that be? And is that the one argument form, the thing where the type drops off, rather than the thing where the type is there? Probably not. Probably it's correct to have the one argument form have the type. But this seems weird to me. So, bio people who are on this call, what is that? I mean, when would that come up, of having an unspecified, we don't know what this is talking about type? We've got a sequence, but we don't know what the sequence is talking about. Does that really come up?
So this is going to actually be quite common, where you'll have a sequence, and then you'll need to guess at what it is. And usually it's quite obvious whether it's going to be amino acid or DNA or RNA, just by the letters, but that you won't kind of tag it as such when you first enter it into the system.
Well, okay, so then one possibility is biosequence automatic, whatever. If it's plainly obvious, it will just go to whatever it is. I mean, if you see PYR or something like that as if you see something like that, you kind of know it's a peptide.
Yeah.
And then,
So,
or potentially inherited, in case you work in a certain context.
Possibly. Yes, either automatic or inherited, but I don't like unspecified as a word. It just seems wrong.
So I guess this was to the case where the user just types in a sequence and then doesn't specify whether it's DNA. What we're saying now that the design should do is, if it's obvious, if there are no conflicts, just pick the right type. If it's not a type that's found, what should it do?
Sorry, say that again.
So the user types in.
You
You know,
type,
they
you type a bunch of Gotic characters. What should happen?
If you say "Hello World" in here,
Yeah.
what does it do? I think it gives a message.
Hmm.
I mean, I don't know, what else could it do? I
And
mean, it could. I don't think having. I mean, it's like having an entity type where you've typed in an entity and the entity isn't one of the entities in the entity type. I think it just gives an error. I mean, I think maintaining something which is a symbolic entity type, symbolic biosequence type, seems like a mistake, especially when we have these things like circularity that are going to affect the operation of other functions.
Mhm.
But I could see it to be useful if we have this mixed ones where there's a piece of DNA, piece of RNA, and there's something in the middle which I haven't clearly identified yet what it is, but I know in the lab there is some kind of macromolecule sitting there.
I think there should be a different form for that. But look, this idea that you've got letters, but I mean, this sounds very weird to me. You've got some letters there, but you don't know what the letters mean. Is that really a thing?
No, you know what they mean, but you have not decided on the name of the type.
No, I understand, but I think Trevor was suggesting that there are random disembodied AGCTQ things lying around where somebody just says, "Here's the sequence." Well, what does the sequence represent? Well, we don't know yet. Does that really happen?
So this happens to me when I'm blasting stuff against the general protein, the database, and I'm going to be lazy and I just want to like run it in BLAST, and then I'll just drop the sequences in, and I don't want to tell BLAST -- DNA or -- protein. I just want to run it. And then it's nice having BLAST kind of just figure that out for me for what it should be thinking this thing is.
I see. Well, I mean, that, I have to say, suggests that the one argument form of biosequence could just do something like that. It seems awkward to have to put the automatic there, but maybe not. I mean, so the suggestion would be if you type biosequence automatic ATTTGC whatever, that would turn into, it would canonicalize probably to biosequence of DNA, etc. Is that right?
And if it's,
of course, it doesn't know that it isn't a circular DNA, but it's going to have to assume that.
And if I guess there are multiple types that it could validly be, maybe it doesn't evaluate, but gives you a message telling you what types are valid. And if no types are valid, it also gives you a message that tells you that.
Yeah, and BLAST will say assuming it's protein, and it will just tell you, it will kind of read that, give you a message.
Right. Are we going to have a control equals mechanism where we can do things like that? Because this programmatic form, I mean, if we say control equals that sequence, we already have support for some of that stuff.
That makes sense.
You can probably do it.
SWSH on our live stream is asking whether they're looking at this stuff and wondering whether they should get the FASTA file for their DNA. I strongly recommend it. I got my whole genome in 2010, and it entertains me a lot. Well, I don't know. I look at pieces of it for random reasons at different times, and if I know anything about genomics, it's because I played with my own genome. And yes, you can find all kinds of stuff from it. Some of it useful.
Here's a potential idea for a circular one, but I'm not sure. That might make more trouble than good. We could use something similar to what we use in real digit and continued fractions, where we have double list to indicate the periodic aka infinite circular part.
Yes.
But it might be more confusing than helpful.
I mean, I can think of other things like continuation characters and so on in the string. It seems like, I mean, we're going to an awful lot of trouble for an awfully weird case. I'm just waiting for the Möbius strip. Are there Möbius strip double-stranded creatures and things? Or is that a silly concept? Probably they don't work. The 3' and 5' don't join up like that.
That's a really funny thing. You could have a single-stranded RNA Möbius strip. I don't know if it's been created.
Fair enough. Okay, but that isn't like the creatures that. Yeah, anyway.
No.
Okay. All right. So this is a list of sub-biosequences. Okay. Fine. With this rather weird thing that we don't fully understand here.
That's right.
So, I mean, maybe we need two different forms. We need an automatic that does the conversion, and we need some kind of biosequence none thing where you can just put any random garbage in there.
Right.
And for
Just
certain
to keep
purposes,
it there and hold it.
right. And it will do nothing. I mean, most operations will just say, I don't know what to do about that.
Right.
Okay. Okay, that's fine. What's that doing? Is that auto-flattened?
That's it could be alternatives. It's either or. So then it's a degenerate case.
Oh, okay. So it's not auto-flattened. And why isn't that then represented like this? Or would that be inconvenient? Is auto-flattening useful or not? In other words, if you're splicing in some, I mean, I can imagine a case where you want to splice in some sequence here, and where that sequence you're splicing in is in a list, and where I would want to just have a list inside a list and have it auto-flatten. But maybe that's just silly. Anybody comment from the biology point of view?
Might be useful to use the list to handle branching as opposed to alternatives. Disambiguate.
Okay. Explain branching. So needless to say, these molecules can branch, but what does that mean? I mean, what do you have to be able to, is it always a tree or could it be an arbitrary graph?
Good question. I think synthetically, the way I've seen it is always as a tree. Like you'll couple a base that has two positions that can continue growing and go from there.
You know, I have to say, it makes me feel like that a better idea for this would be to support this together with it, and this reminds me of NetChain, to support something which is essentially a graph representation here, just like we have NetGraph. I mean, it's a bit weird. I mean, this would be, and again, I don't know how common all this stuff is, but you know, we got a biosequence here to be able to put something in here that represents an arbitrary, I mean, maybe this is completely crazy, but you know, I can imagine, okay, given a molecule, right? A molecule is represented, we got some chemistry folk here, right? Given a molecule, the molecule is represented by a graph, true?
Yes.
Yes.
Yeah.
Okay, so walk me through, if I wanted to stick a graph in here, what would it look like from the point of view of our molecule? So let's imagine I've got a random molecule here. And I say moleculeify that. Why it's taking time, I don't know, but anyway. Okay, so now if I look inside that, am I going to find a graph here? I'll find a big mess here. I'm going to find this list of bonds. I see, I find implicitly a graph by virtue of these numbered bonds.
Correct.
Okay. I am wondering why there's this weird precision thing here. It might be worth looking at, but anyway, no matter here. Maybe that's just life of input form. Okay, so what is the analog of that for arbitrary? I mean, I would assume that these things, I mean, in a principle, you could represent, okay, question, could I turn this into a molecule? In other words, if I were to, this molecule-like representation, what is the correspondence between these two things? Clearly, these are, whatever, bases or whatever, which are whole big clumps of atoms, so to speak. But how should I think about the correspondence between these forms?
So between the biosequence and the molecule, or between the entity and the molecule?
Between the biosequence and the molecule.
Right. So the biosequence is a more convenient form for these large molecules, right? So you don't have to keep track of all the atoms in each one.
I understand.
There's like an implicit attachment point when you talk about like peptides or DNA. It's like which because it's the most naturally found one that it connects to. And that matters in carbohydrates where they naturally branch. There are different attachment points. There's language you use to refer to like where it's being attached on each of the sort of blocks that are going into the sort of metagraph that's larger than that.
But so wait a minute. Carbohydrates is not so. Where do we represent your average long carbohydrate thingy? Is that represented in molecules? Is that represented in biosequence, or where?
No, we were thinking we need something else for this
Yes,
stuff.
we don't have this yet.
And why? Because it's a combination of something which has some kind of graph structure as well as something which is a linear sequence structure, or what?
Well, so I mean, they both have some kind of graph structure, right? I mean, the biosequence is a way of writing a graph, and so the same thing.
But you can't have the adenine, the cytosine, these are the spokes in the DNA, right? You have to how to connect the ladder stuff.
Right. So I mean, that's a chain graph if you write it like that.
For the biosequence, there's a relative small number of elementary building blocks that we want to use. But say for polymers, there is a large number of elementary building blocks.
I see. And in general, you could imagine, am I right, that there's essentially a graph's worth of, the connection of those building blocks could be an arbitrary graph in a way that here we're representing individual atoms with bonds, but we could be representing subunits of a polymeric molecule or something,
right.
with connections.
And potentially even hierarchically.
Right.
Right. I mean, you could imagine having a macromolecule where the first argument was a paired like atom or molecules plus attachment rules, right? So a list of lists, each one has a molecule and a list of attachment points.
But
and then in the second argument, you have bonds between the types that were specified in the first argument.
So you're imagining that we might have a macromolecule wrapper in addition to the biosequence wrapper.
Yes, we had discussed this in the biochemistry group, and we saw.
So if macromolecule were representing an arbitrary polymer, which had 3 different repeating monomers, right? So the first argument could list those monomers and the amount of the information needed to add them to an existing chain, and then the second argument would be how those first those repeating units are laid out in the graph structure.
So how would that correspond to, I mean, in the case of, so in the case of net graph, what we're doing there is we've got a graph, but so here, these are the supply of monomers, A, B, and C, let's say. Whereas in net graph, and we might use A 1,000 times in the macromolecule, right? Whereas in the net graph, we are, what are we doing? We're referring, let's see, what determines the, I mean, there are 2 different things. Which monomer are we talking about, and what is its node number in the graph, if that makes sense? Well, anyway, okay. Let me ask another question. I assume we want to say a biosequence with an uppercase S. Yes, no? Any comments on this? Probably we want it with a capital S. Anybody got any thoughts on that? Seemed like a trivial question, but we're going to see this a gazillion times, so we might as well get it right. Anybody think it should be with a lowercase s? Anybody think it should be with a capital S?
I prefer the biosequence as written.
Okay, all right,
Yeah,
fine.
I do
We've got
too.
at least one opinion. So okay, good. 2 opinions. All right. Okay.
But it was the case where a molecule is on the surface or something, which we also can describe.
Sorry, one more time.
This is about molecules in their physical location, like random molecules that are sitting on a surface. I think that's getting into, yet more. Yeah, right. It's
We have to eventually have to get to solid states, but this is a totally different thing.
Right. Well, and I mean, I assume that these biomolecules also have the feature that they, they are in an environment. They're in some aqueous solution or something, and that probably matters to them. But we're not recording that fact now. True, not true, irrelevant, relevant.
But that's already true for a single little molecule.
Right, fair enough. All right, what's this? Additional types of biosequences. Okay.
It's for you how you write a custom one.
Right. But we got, okay, so one thing we talked about is the alphabet mapping, what these things actually mean.
That's right. If looking at the sequence cap, you'll see an example of that.
I would just call that term chemical association. I really think that's overly fancy. I mean, I would call it
Chemical realization.
Or something. But I mean, sequence terms, I mean, that seems very weird. I mean, that's like sequence terms as in the sum of 1 over n squared, the sequence terms of the, 1 half, whatever. What are some standard names for these?
Alphabet.
Alphabet, yeah.
Okay, well, alphabet will be fine by me. I mean, the
Sounds great.
Yeah, that's
okay.
standard.
Okay. Then this thing caption, isn't that a standardly named thing in an entity? What's that called? Isn't that called label?
Tony uses label for he maps it to something else.
Okay.
And
So this is short. What is that? Short name or something?
Or that's the picture that they want to use.
Summary box.
Okay, so that's
It's a type. It's a type.
No, it's not. It's the icon, literally, and it would be a picture.
Yeah.
Correct?
Yeah.
Okay.
Or if you want to you don't want always to supply one. That's why we should allow reusing what we have.
Fine. Okay, but so the DNA is that's a shorthand meaning use the DNA picture.
Correct.
That's right.
Okay. What should this be called? Complement rules?
That makes sense.
Reasonable? There are two-way rules.
It also takes the rules in the single direction. That's just a shorthand.
And presumably can take list pairs as well.
Yep.
Okay. I don't know what the source means.
Who published this one?
That should surely be an external
Yes.
identifier.
It will be.
I think that should be called something different, like bibliographic source or something.
Sounds good.
I don't know what. Okay, what is a sequence cap? Wait a minute. Why is this? So this is just examples of two different kinds of entities, or this is the sequence cap of a Hachimoji DNA.
They're two different examples.
Okay, fine. Okay, terrific. Okay. Okay, so then we just need, I mean, we could call that alphabet rules.
Alphabet map.
Okay. Okay. Okay, so this will sit as a biosequence of biosequences. Is that correct?
That's right.
It will not simplify in any way. And the canonical form, but if I had a DNA inside, if this said biosequence of DNA, it would just flatten to a single DNA. Is that a true statement?
It should. If it doesn't, I'll make sure that it does.
Okay, but is that biologically correct, guys? In other words, if you splice two DNAs together, you just get a DNA, and the splice point is now irrelevant. Is that a true statement?
Sure.
Yeah.
Okay. Okay. Sequence string, fine. Now, why are these being called chemicals? Are they molecules or are they something else?
They're chemical entities.
Okay, all right, fine. Okay, now why is this list not a list, so to speak? Why is this being, I mean, I understand this is one of these biosequence of biosequences, but is that the right thing to do there? Probably is.
It's the only possible way, or? I mean, we want to preserve the nesting structure.
Fair enough, but notice that in the case of the sequence string case, you're not really preserving the nesting structure. Well, I suppose you are by having those as separate. But wait a minute. No, this is a tree. This is a linear sequence of pieces. Am I confusing something? This is only one level. There's only one distinguishing level here, whereas here you could have multiple levels of nesting.
But if the first example would be multiple deep, then this one would be too. I'm confused.
Would be lists of lists.
Yeah. Okay,
fine. Yeah, I guess you're right. So the only difference is that the bottom level of list here is turned into a flattened string.
You can join characters, you can't join chemicals.
Right.
Isn't this why we want the alternatives thing, to get rid of the ambiguity?
Yeah, this one should be an alternatives right here.
Yeah, so that's not gonna. Okay, so fine. So we agree that the pairs as, I don't even know where I was. Oh my gosh, I lost my place. So
I'm a little bit worried somehow with this alternative. I feel like the alternative is more a pattern rather than a representation of an unknown fact.
Well, it is a pattern. And if you did a match against it, it would.
That's exactly my point. If you start doing matches against it, you must go one level up.
Let's look at that in a minute. Okay, but it's clearly not going to work to have a sublist there representing alternatives.
Definitely. I was thinking on the new head.
Oh God, let's try and avoid that if we can. Okay. Yeah, I mean okay, categorical uncertainty is the general category here, right?
Correct.
And what are we doing for other forms of categorical uncertainty, like in random choice or something? In random choice, we just have a list. But it's an interesting question. What I think we have used alternatives as a representation of categorical uncertainty in other cases as well.
In outputs?
Not sure. Not sure. I thought we were thinking about that for some quantum mechanics thing recently.
But then it becomes hard to pattern match against it.
Okay, let's think about the match here. So you're saying this is an output, and you ask the question, does this string match a string that contains one of the particular things in it? Right. And your argument is the match is the wrong way around then.
Exactly.
Humph.
This is not a pattern. Alternative is a pattern construct, not a uncertainty representation. Then I would be more in favor of overloading OR to work on non-Booleans.
Oh gosh. That's a funky idea.
It would be really consistent with colloquial English.
That would be very hard to understand. I mean, so one of them is the pattern construct, and one of them is what would normally be a control flow construct.
Yeah.
Ugh. Well, okay, we're going to come back to this. Okay, we've got more to cover here. I agree. There's an unsolved problem here. Okay, we're finally on to section 2 here. I wonder if it's as long as section 1. Okay, find biosequence features. The heck is that?
This was your original example a couple of months ago.
What is it? Find. Why is it this way round? Why isn't it on the Why isn't it the other way around? You're finding in the human this biosequence.
This is very similar to, I guess, yeah, in this case, it should be the other way around. Although then with the SNPs, that's, yeah, the ordering doesn't kind of really match between the two.
Hold on. First question is, in the human, I mean, this is kind of an elision anyway to say human, because you're really talking about the human's DNA. In, the reference human sequence, find, okay, so this is a kind of complicated thing. All right, so here, I don't understand this at all. I'm sorry, I don't understand. The biosequence position should be a biosequence position. Why does it give 552 after it? Isn't it a sequence position?
So in chromosome 16, at this particular feature, that at 167 to 31, that corresponds to position 552 on the biosequence that was provided.
Ay. No, I don't understand this at all. I mean, I don't understand. I thought what we were doing here was doing essentially a sequence position to try to find that biosequence in the human genome and then return the result of that.
Yeah. So the position 552 of the given biosequence is found on the human genome in chromosome 16 at position 167 to 231.
Oh, but that doesn't make any sense. So you're saying that complete sequence was not found. It was only the subsequence that starts at, I assume, because I mean, to find a single base pair is hardly a right home event.
Right. The challenge here is that if there are all kinds of other SNPs in the sequence, then we have to find a place to hone in on. And this gives us one position where we can say,
I completely don't understand. Okay. So, times when like I match some sequence against my genome, okay, I just find some sequence positions on the genome that match it, right? Which I would assume would be a biosequence position. I don't understand this. I mean, you're either matching it or you're not matching it. What am I missing? And I don't understand why this thing here, I don't understand why you're returning something which has this in it. This doesn't make any sense to me. I mean, I would assume that you find the feature on the genome and it returns as a biosequence position. What am I missing? Somebody. By the way, somebody's typing is very, very audible. Okay, am I missing something here?
You might be right. I thought I had understood it why it was good, but right now I follow your argumentation.
Yeah. Okay, well, let's keep going. So this is asking, this doesn't make any sense. This should be human SNPs. Okay, this is a different way around. This is, in this biosequence, where is that SNP, right?
Right.
Okay, but this biosequence, by the way, who was typing might want to put themselves
I think that's Jeff.
Okay. Okay, so in this case, it's find this SNP in this biosequence. This biosequence is anchored to the original human genome. Is that correct? And what is this? What is this thing?
No, we don't know.
We don't know. It's just some random sequence. Okay, so then I don't understand this here. I don't understand this at all, because this SNP is anchored to the human genome. This RS designation,
right?
Something which is
this does the work of then determining if that SNP, if it matches the sequence to that region of the genome, sees if it aligns, and then if it does, finds the position inside the biosequence where that SNP is found. So it does the work of doing all of that alignment and so forth.
Okay, but what if that biosequence, like if it's 1,000 base pairs long, and you can see the flanking sequences on the human genome of that SNP, and they match in 900 of the positions, okay? What does it do? What is it trying to do there? In other words, at what matching level will it decide, yes, I found...
In that instance, it will fail because it wouldn't be able to align the position that was given uniquely. Only if it finds uniquely along where it is on the human genome will it then attempt to find a SNP result.
Okay, a bunch of people on our live stream are pointing out the fuzzy matching is one of the issues that comes up here, and for sure. I mean, I don't know how that's represented in this.
That's right. So this is a case where it's competitive. So you're given, you have some sequence. It may have some number of SNPs in it already, so it's not going to align.
No, but I'm asking the more basic question. Even that first example there, finding a particular biosequence in the human genome, right?
Right.
The best case for that is, so that biosequence might have pattern variables in it, might have ends and things in it. It might also be the case that, oh, you know, it's a length 2000 sequence, and oh, it's a really close match for this place, but it isn't a precise match. So how do we deal with that?
So it works because it's competitive. It's not comparing it against if it's a perfect match. It's comparing it against all other available matches. So it's slowly building evidence of...
Okay, well, fine, but is it going to return one of them, or is it going to return... I mean, like, for example, I've done this with genes of mine that have, you know, where there are two very similar genes, and I try and match, I try and find some random SNP by looking at flanking sequences, and it's confused which, you know, I'm confused which gene it's actually, you know, which thing it's actually on. Yeah, so what does it do in this case?
So either it will overrule one or the other, or it will fail. It always, this particular method always has to find the best match given some considerable evidence.
But so it could, in principle, there could be two identically weighted matches in principle.
Right.
But you're choosing to return one, and for some reason it's encrusted in multiple levels of list with random numbers there.
No, if it's two and it's close, it'll fail. It's only if it can find one distinct one that where the positions are better compared to some other one will give you a result.
Although that's not necessarily desirable. I mean, that may be what this particular
that's fair.
does.
I mean, you know, that's
so we might want to design something which has the possibility of doing something better than that. But okay, so I'm trying to understand here. Okay, so let's just understand. Okay, hold on. Let me just see what's happening here. So wow. No, that's the wrong way around. You're finding it in the human genome, right? This is the thing you're looking for. So it's like position in human genome of this, and that should be a third argument saying what you actually want to return. I think. Does that make sense?
I think we did that last time, and then the similarity to text cases was pointed out.
Okay, but this is the wrong way around, because it's not in text cases. It is in this. It's the wrong way around. I mean, you know, you're confusing yourself because you've got the SNPs thing, but the SNPs thing really just represents the collection of human SNPs, doesn't it? It's not a designator saying find me. It's saying find me something from the SNPs database that matches. I mean, and the confusion here is that the SNP is just that one location, but it isn't really. It's really it plus a bunch of data about the flanking sequences, isn't it?
You know, I disagree. The flanking sequences are used here to make sure that the SNP is actually in discovered sequence. Yeah. What's going on here is, to my mind, like as though this were some other entity class inside of text cases. We're saying, find me all of the foods inside of this sentence. Find me all
but
of
we're
the
not
SNPs
finding all the
inside of
humans.
this sequence.
We're not finding all the humans inside that
So
sequence.
it is true. So I think that the species specification was a poor choice. I think that the genes case where we're finding the human genes inside of the given sequence makes sense. I think finding the SNPs inside the sequence made sense. And I went with the human species specification out of uniformity, but I can see that that is confusing.
Okay,
but,
so
but
what
we
you
don't want an entity for the human genome, because what would it be different than the human it represents?
Okay, so hold on a second. So what this is really confusing. Okay, this is saying, representing this as the human genome. Oh boy. But this is, again, the opposite way around than text cases. Right? Text cases, you give the text you're looking in, and then you give what you're looking for. Right? In this case, what you're looking in is the whole human genome, and what you're looking for is this biosequence that you fed in. Right? So it's the wrong way around. And then what you're returning is a separate issue. Let's keep going a little bit here. Okay. Oh boy, oh boy, oh boy. We have a lot of stuff to go through here. All right, so this is definitely wrong. So find biosequence position. I am so freaking confused by this. I mean, this is, you're choosing to return two things in your symbolic representation of what's to be returned, the feature and the position here. I don't understand why that sequence is why that sequence position isn't the sequence position. Is the sequence position a single place?
Yes, the sequence position is a single place for the purposes of.
But then I'm totally confused again, because then what you're looking for is, where is the beginning of the sequence?
No, the sequence position is a range.
But it's not. In none of these examples is it a range. Yet this biosequence, I can plainly see, because it's sitting right there, it's a long sequence length 842.
So the way I guess I interpret it is this position corresponds in the given biosequence 552 corresponds exactly to the DNA sequence position on this chromosome 16. And if you kind of it could have been arranged, this just happened to be kind of an anchoring position that was found that's useful for.
Well, I think it's crazy, because I think that if you're going to say, where is this biosequence on the human genome, the answer is it's a range here. Right? If you're going to say, where is the start position or something, I don't even know if that means anything, particularly with reverse sequences and things like this. It just seems wrong. Whereas this thing here seems like a better thing to be returning than just these raw numbers. I mean, again, I like the bio folk to opine on whether the raw numbers are useful. So, I mean, when I've done this on my own genome, I often just use raw numbers, but that's just because I'm lazy. But it seems like a bad idea to use raw numbers. I mean, isn't that what the whole RS thing is all about, not using raw numbers?
I'm used to raw numbers plus a reference genome when I think about these matchings, which this seems to align with. And then very commonly, 2 steps back of getting a ranked list of all of the matches that come up.
Okay, so biosequence position should be like a lat long on the Earth, relative to a certain datum. It means this thing, and that's what this should be concentrating on. I would have thought it's a biosequence position.
So if you go ahead and click on the plus.
Okay, but that's just it. This is not a good way to say it. I mean, it isn't. I don't care what the feature is. Well, this is saying, okay, the first thing this should say, in my opinion, is this is a sequence position in the human genome, right? That's the
that's
first
good.
thing
Yep.
to say. Then, and it can say it's human genome, you know, and then it can have a little gray version, the assembly 36.3 or 36.3 or whatever it is. Okay, then this is what feature of the human genome this happens to correspond to, right? Well,
That's right.
actually, the first thing to say is this 552 is presumably a coordinate. Well, it's a coordinate on the chromosome 16, isn't it?
The 552 is a coordinate on the given biosequence, which maps to
that. Okay, that's just crazy. That's just crazy in my view. Look, if you've got a biosequence here, make this biosequence position, or maybe it's a biosequence segment or something, just say, where does that correspond to on the human genome? Don't have this hacky thing. I mean, people have probably done this because they don't have a way to represent some symbolic sequence thing. Why would we want this? Why don't we just say this symbolic thing represents the segment of the human genome that happens to lie in chromosome 16? It's useful to know that, rather than just sort of randomly saying we don't know where it is. And then say, with respect to chromosome 16, it lies at these coordinate positions.
That would be my expectation, that this would just be 167, 236.
A single thing, or it would be the span of
that would be the span.
Okay, fine. Okay, I think that's what it should be doing. I don't know what this is doing. I don't think this is useful. This is aligning this number with that position in this thing. Just seems completely backwards and confusing.
Can I ask, why is it 552 rather than, or just so I can understand that?
Right.
Yeah.
It's for the purposes of robustness, I tested this with some SNPs at the beginning, so it doesn't actually align to the beginning. This is the best position overall for the entire sequence.
I see. So you found a canonical SNP that's at 167, 231, that it's a labeled entity, and it's relative to that SNP.
Right.
Okay.
So you're saying that position 552 in this DNA sequence aligns with 167, 231 in this chromosome. Is that
exactly
correct?
Right? Yep.
Okay, but I think that's not what you should be saying here. I think the primary thing you should be saying is, I mean, if this is a match that's worth its salt, so to speak, then I'm now confused by what that might mean biochemically, but never mind. Then you should be saying this sequence lays down on here, and maybe you say it's anchored to this point.
Mm-hmm.
As a sort of a footnote. That's how it found that match. That's somewhere down here below the fold as more of a detail.
Oh, that makes sense. Yeah.
Trevor, does that make sense to you?
Yes, that does. And then commonly, kind of, if you drill down in BLAST or whatever, what's really useful is you get a little alignment showing kind of the seed of the sequence that you gave and kind of how that aligns to the other thing, and that alignment will have positions annotated on it, and that just makes things crystal clear of what actually happened there.
Fair enough. So it's an iconic thing that maybe gives some color coding of how good the match was away from that seed or something.
Mm-hmm.
Then we would have a sub-value for the biosequence position that potentially gives you nice graphics.
Exactly. Yes. Okay, but what's the difference then between biosequence position and biosequence segment?
So it's that position was at in reference to a single position, while segment is in reference to a range. So
I'm going to claim that position doesn't make any sense. That we don't even need it.
I'm open to that idea. Yeah, let's go in that direction and take it out and just keep sequence position with this new compact form. Yeah.
Okay. All right. So then. Okay, so we got a bunch of details about what we return. I claim that should be a third argument.
Sounds good.
And I claim that the default return has none of this hair in it. It's just this one object. And I don't understand why this is why this has got features in it. I mean, who cares? Why don't we just say find biosequence? And you're just saying find biosequence of, so then it would be find biosequence, and then it's big, small, and then features if you want to. And that would return a biosequence segment or something. And a biosequence segment would contain inside it a biosequence or a reference. It either has its own biosequence, if it's a de novo, if it's a sort of a constructed thing, or that biosequence that's inside it is like the human genome or something. Does that make sense?
Yeah, there's a reference feature if it's available. Yeah, that makes sense.
Well, if there isn't a reference feature, right, then you've got an actual, then you've got essentially a reference, which is in our world is just a copy. I mean, it isn't really a copy; it's a pointer in memory somewhere, but of the original sequence that's just sitting there.
Right.
Okay. And then, for example, in that case, if you were to try and splice a biosequence segment, so there's a question then, if you have something which has okay, here's a question. If I've got a biosequence segment of the human genome build 36, and I try and splice it into build 37, that's just a meaningless thing, right? I mean, if I'm to say, because I can imagine a very common thing is I've got a biosequence segment and I've got my particular personal DNA and I want my version of that segment. How would I get that? In other words, this segment represents some range of positions on the genome, but I want to take that cookie cutter, so to speak, and cut out my version of that from my personal genome. How would I do that? And is that a reasonable operation? Biofolk, is that a reasonable operation?
So I'm trying to parse.
Okay. So what I'm saying is, if what comes out here, we say find biosequence, okay? And what I'm saying is I want to find, okay, so big, let me try this for a second, okay? So let's say I've noticed that every time there's a ATTTTT something or other, and some long sequence of somethings anywhere in the human genome, that the human in question will be bad-tempered, let's say. But then what I do is I use this to find out where on the human genome does that sequence in the reference genome, where does that occur? And then I want to say that place where it occurs on the human genome, now on Bob's genome, I want to find out what's at that position. Is that a reasonable operation?
Yes, definitely. And usually that's talked about in terms of SNPs as like the core thing, and then there'll be DB SNP and databases of particular SNPs that will actually have names to them. That makes that really easy. But then you'll have some larger kind of rearrangements and things where you'll need some more kind of bespoke intervals and so forth.
Right. But so okay. So I mean, this extractor, this is like a position specifier. Yeah. I mean, maybe the right thing to do is to call this biosequence position, but have it often be a range. Because then what we would say is extract from Bob's genome this biosequence position, which might be specified by one of those RS snippy things. Or it might be specified by something that was a raw set of coordinates on build 36 or something like this.
That makes sense to me.
Okay, so this is probably Okay, we'll try that for now. Okay, let's keep going. I haven't a clue what this means. I mean, I know what it means, you know.
So I get the second example may help.
No, I understand what this means. I mean, I know what the sort of biologically means, but right. So we get rid of these numbers here because those
Right.
have all been absorbed.
Yep.
And these then become this is reasonable. I mean, this is just saying biosequence position gets that probability, et cetera, et cetera, et cetera. That
Yep.
seems fine. I mean, that's very similar to what we would return from the machine learning system.
Right.
Is it also an association in that case?
Yes, this is identical to that case.
Okay, fine. Looks good to me. Okay. Oh my.
These we're going to change, of course.
Okay, but that's the innards of a biosequence position, which probably
Right.
should be in an association. I wouldn't have a naked thing like that, probably.
That makes sense, given that we're giving back as data set too. I can also take the data set.
Yeah, why not? But that will have ranges in it.
That's
And
right.
I mean, and by the way, this plus, I can't imagine why you wouldn't canonicalize that with a capital P.
Makes sense, yeah.
Okay. Okay, what is biosequence segment and how does it differ from biosequence position as we've now defined it?
Oh, I think they'll become one thing now that we're always talking ranges. That'll all become biosequence position.
Okay. What about biosequence mutation?
This will also then fold in the biosequence and the biosequence position, but otherwise this is, you know.
Okay, so what this is representing, okay, let me understand what this is representing. So this is saying, I want to find out, how do you get to one of these biosequence mutations?
If you find a SNP, if you try and find SNPs.
So okay, so one thing you can do is to find a small and a big, for example. And yeah, by the way, Brian on our live stream is asking the obviously reasonable question. If I just search for AAA on the human genome, there are gazillions of them. I don't just want to find one.
Right. So
and we already do that. I mean, for the, you know, for the case of non-BLAST type things, for the case of pure literal match, we already, you know, we have this suffix tree thingy that does that.
Yeah, we already have genome lookup for that. That's right.
Right, but I don't understand. In this case, being able to do find biosequence, it's a little confusing of that versus genome lookup, but find biosequence, if I just ask for AAA, I better find a gazillion biosequence positions.
It'll find a gazillion biosequence positions. Say we can't define. What it'll do now, it'll fail. It'll say we couldn't find a unique position.
That's not good enough. That's silly, because all of those will have the same probability. It'll be 1, right? So that's just not good enough to have it just fail. Right? If there are multiple matches, it should return multiple matches, and we need a criterion. Okay, so okay, this is now a biology question again. I mean, the thing is returning probabilities of matches, correct? But there's a point at which there really are multiple matches, and the loser probability just, you know, there's a 0.99 and a 0.97 and a 0.94, and then there's a 0.26. What should we do? I would argue we should have an acceptance threshold, which is what we do in machine learning stuff. And above that acceptance threshold, we should return them.
That's how BLAST works, where there's an eval. They have this like pseudo p-value thing. They call an e-value, and then you get everything kind of above the line.
I am depressed that I keep on reinventing things that already exist. Well, it's probably a good sign, actually. So that seems like the right way to do it. Is there a problem with that, John?
No, we could do it that way. Yeah.
Okay, so I would use probably the acceptance threshold option that exists in, if I'm not misremembering it. Does anybody here remember what the, yeah, there we go. Acceptance threshold is an option. That's the thing. It's used for exactly the same purpose.
Great.
And eventually we'll be able to merge these things with never mind. We'll be able to take the genomic version for better or worse. Okay, so now what is biosequence mutation then? So biosequence mutation is conveniently telling me that, I mean, this is the question of, you know, which SNP which SNP did Bob have, so to speak? Right?
Right. It's which SNP fully specified on what, according to what reference?
I understand, but I mean, I'm asking the question that return. So one thing is you're searching for a small and a big. One small you could search for is a SNP, which is this kind of pseudo single position, right?
Right.
And in a sense, what you're going to get out, see, here's a confusing thing. When I get out a biosequence segment, the fact is there are definite values there, right? I don't really need this because in a sense, you're not happening to tell me when I get out that biosequence DNA sequence position, right? You're telling me it's feature position, blah, 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 blah. But the fact is, at that position is a T, for example.
Right.
So, I mean, the fact that you didn't tell me it was a T there is just silly, in my view. In other words, this biosequence segment, and I'm also very confused by why the biosequence segment is any different from biosequence itself, but we're going to get back to that in a second. Okay, because it seems to me biosequence segment is just a nested instance of a biosequence, because a biosequence is saying, maybe I'm wrong here. A biosequence,
I think they're different.
Yeah, okay, I may be wrong.
One is the context and the other is the actual presence of an object.
Right, I mean, a biosequence has its base is the world of all DNAs, and then it is a particular instance of that. In this case, a biosequence position, its base, so to speak, is a particular biosequence. And this is a specific location on that biosequence. Correct?
I think so.
Okay. But that specific location has multiple attributes. It is because it is relative to that specific biosequence, it not only has a position in that biosequence, it has a value in that biosequence. Correct?
Yes.
Okay. So it's not obvious to me that it should be called biosequence position. It should be called what it is. Maybe it's biosequence segment again. And what it then returns in it is it's this sequence of positions and it's this actual set of values. And so you want to get the SNP out, which SNP Bob has. You simply ask what values that segment has.
So there's the values of the reference, and then the values of the biosequence that were provided.
Okay.
Now both are going to be represented here, and so there are accessors for both. But absolutely.
Okay, fine. But then okay, then that's a good thing to have. And even in this thing, you might want to, even above the fold, you might want to be showing, okay, so what are the things that you need to know, and what should this thing be called? Should it be called a biosequence segment, or should it be called something different? So, okay, biosequence segment, let's assume it's is that the best name, or is it biosequence location or something? Well, segment is okay. Okay, biosequence location segment. Then it's got the base biosequence, right? That's the thing that it is relative to. And then it's got where this one is. Well, I mean, I'm making this up because it isn't really the right thing, but it's location coordinates or whatever it is, coordinates. Then it's content essentially, and then it's reference content roughly. Correct? And then it's actually got another thing as well, because that base sequence might be the human genome. Beyond coordinates, it's also got to say it's on chromosome 16 and so on, right?
Right. Now, does it have to, if we're talking about finding genes, does it have to, it probably has to have both sets of coordinates for if the gene overlaps the sequence that was given. So that we're talking about kind of two different sequences, subsequences.
Okay, so let's just understand what this is. So a biosequence segment, the base sequence there is either an actual sequence that you derived from your from a sequencing machine or something that might be the latest bizarre bacterium that produces chocolate or something, and but it isn't a reference sequence at all. It's just an actual biosequence that you got, right? That's one thing that could be in that first argument. Or it could be a symbolically referenced the human genome. Is that correct? Am I confused here? But then if it's the symbolically referenced the human genome, you've got to say things like build numbers and so on there. And then you're saying, relative to that base sequence, you are defining a certain segment in that base sequence.
That's right. We as it's an output of this fine biosequences, there's both the coordinate content of that was either the thing from the lab or the reference sequence, and also the coordinates with respect to the sequence that we provided.
Well, wait a second. Okay. Let's go over this again. Okay. Let's say that the thing we're searching for is a gene. Okay.
Yep.
I don't understand why that gene doesn't essentially read across a particular part of the base sequence.
So the challenge is that somebody gave us an arbitrary biosequence that we found, and we now need to say, given that we know that we found this gene, how does that overlap? How does that refer to? How does the gene refer to the sequence that we gave?
No, no, this is muddled. Look, either, okay. One thing you might do is say, I've got the sequence, AGB, AGTT, whatever it is, right? I want to run that sequence against every organism in my collection. And it's going to come back with a bunch of biosequence segments.
Right.
And so the way that would look is you'd map, what were we calling it? What were we calling that thing?
Fine biosequence, I think.
Which is itself totally confusing because we're not finding a biosequence. We're finding a biosequence segment if we're finding anything.
Fine biosequence segment is fine. Yeah, that'd be great.
Well, I don't think it's right. I don't think this is right. But anyway, let's assume we did that, okay? So then we say map that over, and then what's here is a biosequence, right? Pure biosequence. No reference, no, it's just saying it's DNA, it's this thing here.
Right.
Okay, so now I'm mapping this over, let's say, pig, goat, antelope. Okay? And then what I expect to get back is, presumably, is biosequence segments in which this says is the pig reference sequence. Okay, however that's referred to, and I think it needs to be decorated because you need to say, well, somewhere you need to tell it that I want build 17 of the pig reference sequence or something.
Mhm,
Right?
mhm, Yep.
And this, if I'm mapping it over the pig entity here, that's not telling it that. Okay. So then here, then it's got to go through all this jazz of, in relative to the pig reference sequence, where is the sequence that I gave you? Right? Coordinates, whatever it is, whatever. Right? Now that's what I think that biosequence segment is. Now, if somebody then says there's a resolver for that says, what is the sequence from that sequence string, let's say? Or alternatively, what is the biosequence from that? Because the biosequence, which in this case was rather trivial because we just looked for it, but if that biosequence had a pattern in it, it wouldn't be quite so trivial to say which actual one did we find. You see what I'm saying?
Yes.
So what am I missing here? So let's imagine, what are some other cases here? So we say, another case you're talking about is find biosequence segment of, in the pig. Well, we've already got in the pig, find this. So then another case would be find it in this biosequence, in my custom biosequence, which might be Bob's DNA, or my DNA for that matter. SWDNA here. Okay, so we've got this custom sequence here, and I'm going to, and then we want to find the position. So we might want to find the position of the SNP with an entity, SNP entity here. And then some random RS, whatever it is, right here. Am I making sense? And then what we want to get back there is, what are the particular coordinates on my DNA that correspond to that SNP? And what is the actual biosequence that I have at that SNP, right? And that actual biosequence is, I've got to see, if I was smarter, I would have a T or something. Does that make sense?
This is good so far. The case now I'm worried about is where the features are about the same size, where you have a biosequence and you're looking for if a particular gene is inside of it or overlaps with it or something like that.
Okay, well, let's look at that. Okay. So what you've got there is you've got something where you are saying that you are prepared to allow, I mean, if we were doing string matching, your string match would simply fail if one end of the string wasn't if the string only matched, you get what I'm saying?
Yes.
Only half the string matched, it would simply fail, right?
Right.
So what you're asking for is a match where, your reference ref sequence and your, sequence you're searching for, where the sequence you're searching for is not contained in the reference sequence, correct?
That's right. It might not. It might just overlap.
Okay. Okay. So biofolk, are those two things considered the same in biology? Sequence is contained in the reference sequence, or sequence overlaps the reference sequence?
I think so.
Okay.
A small note here that internally for what we're doing, we generally work with FASTA files and VCF files. FASTA being just a sequence, which would be, I guess, be biosequence in this case.
Yep.
And also work with variant call files, which is then basically you always need to be working with a reference and a VCF, and the VCF tells you how you need to transform the reference to get to what you have. And that doesn't have to be the case. You're doing much more, but that's kind of what internally we found useful.
Okay, so in other words, yet again, the concepts that we're inventing here have already been invented. That is, the VCF thing is an anchored, the FASTA file, right? Like, I got my mitochondria first years and years ago, and it just came as a blump. Here's a bunch of, here's a sequence. And that was all the FASTA file really had in it, apart from a little bit of random metadata. Whereas the VCF thing, you're saying it's an anchored thing relative to a reference sequence.
Exactly. That if you got your genome sequence today, they could hand you back rather than 3 billion base pair FASTA, they could hand you back a roughly 100,000 line VCF that would tell you for each of these SNPs what do you have relative to the reference. Where you
it's longer than that, right? I mean, don't you have like 700,000 de novo SNPs?
There you go. Okay, sorry. You know the number better than I do. 700,000.
Yeah, okay. Well, but that's the de novo SNPs. That's the SNPs that you have that have never been seen in a database. The number of SNPs you have relative, anyway, it doesn't matter, but it's a longer thing. But I get the point. It's much shorter than the 6 billion. Okay, so now you've got this case of the overlapping thing. Well, I don't really think that's a huge problem. I mean, you basically are there's some coordinate thing you have to specify here. And, I don't know, it's some footwork that we're not going to be able to do here that will say that.
But, I see. Yeah. I think it's that. I understand.
Right. But I don't really like these names at all. But I think the concept is, what's that?
No, the names are now no longer correspond to what we've discussed. That's true.
Okay. Okay. SWSH on our live stream is pointing out about analogies to temporal data instead of common time segments have. Yeah. So what happens in the case of multiple reads? The output from a sequencer does not contain just a single sequence value at every position.
Oh, yeah.
Yeah, so there's this whole assembly problem, base variant calling problem that you'd have to go through from your kind of pile of raw reads to this consensus genome that you're dealing with here.
Right, but I mean, if we wanted to represent the raw reads, I guess those are usually compressed into saying it's a C or a G or something, rather than ever showing you the whole stack. Okay.
Yeah, the VCF will work after you've done assembly is the sort of thing that people use for this, where it would represent of your raw reads, 40 out of your 100 at this position are calling it as this thing.
So that's again something that we're not able to represent here, which we would like to be able to represent. The different probabilities for each. I mean, one could imagine in the biosequence, we could imagine something like this, where we have, A, G, C, T, and then we could say something like A 0.6, G 0.4 at that position, etc.
But wouldn't this not be?
What?
I was more. Yeah, I was more thinking like typically we have weights goes to sync.
Fair
but
enough.
if it's sparse, of course it doesn't work.
That's a separate issue. I think this is probably the better way to do this, because this will be better for, by the time you get to amino acids and things where it's quite a sparse story. But the question now is, so a question from a live stream is, do you really have to represent? So as you say, individual humans differ only by millions of base pairs, you don't have to specify all the billions. And that's like saying, so if you're throwing around genomes, we don't want to have everybody's genome completely represented in the system. So how do we deal with that? Can we have a biosequence that is a relative biosequence, so to speak? In other words, right now you have a type here, but you could imagine something which is a biosequence in which there's a base biosequence here and which there are diffs here.
Mm-hmm. Yeah.
And that would be analogous to the VCF file.
Yeah, that's a cool idea.
And then as far as manipulating these things are concerned, we would have to make it so it doesn't make a difference which one you have. But this would be, given that you're dealing with a human, you might as well represent it this way. If you're dealing with an unknown Martian bacterium, then you don't have a choice. You just have to represent the raw thing. But now, in this model where you've got diffs relative to a base, this is making biosequence segment look an awful lot like biosequence, where the only difference is that you're saying, if it's a custom base, you might say the default for biosequence is it's the whole base, if you understand what I'm saying, the whole base sequence. But an alternative is it's just this coordinate position. In addition to diffs, you can be doing that as a selector. So you say it's just this coordinate position relative to this base sequence, right? But if you're simply typing it in as biosequence of AGCT, whatever it is, then, I mean, I don't completely understand the base space of having DNA versus, and I could even imagine that the real correct form is something more like biosequence of, something like DNA arrow base diffs. I mean, although, this could just be a single entity because, it might just be the human genome, which we know is DNA. I just think there may be a useful factoring of the base type of thing you're dealing with relative to the actual. Okay, so that then instead of biosequence of DNA, comma, yeah, I don't know. It might be useful. Look, it may be useful, but then you've got these mixed things. I don't know. I'm not sure. Not sure how to do that. But whether you want instead biosequence of DNA, the base sequence, and then the diffs here. And that's the only data structure you're dealing with. And those diffs could mean I just want this part, or they could mean here are some changes relative to the base. Does that make sense? And then the accessors would, so there's clearly an accessor which would turn this into the pure sequence string, and that would resolve both the part extraction and the diffs, right? And the diffs, presumably, I would assume would be, well, I would assume that they're indel. I don't really know how that works, and I don't know how you store. So in VCF, presumably, is it storing indels, substitutions and indels?
I don't know. It should, but I don't. It might not. It should.
Well, but that's not a trivial thing, because a deletion at the beginning is going to shift everything down. So there's probably a much smarter way to store these things that involves, you don't want to have to, in other words, you have to do essentially a prefix type thing where you're going through, it's like a fold listy type thing, where you're starting at the beginning and you're saying, oh, I've seen this number of deletions, therefore my coordinates are off by this amount at this point. See what I'm saying? So
yeah, I think that's what we're saying.
I mean, it would be rather inconvenient if you say, oh, this thing that has these diffs, now I want position 100,000. Oops, I have to go resolve the diffs before I know where position 100,000 is. There's got to be a better data structure than that for it. Where you kind of, there's got to be a data structure in which you basically are storing, I don't know. I don't know. Anyway, okay. Okay, we are beginning to run out of time here, and oh my gosh, look at this. Oof. Well, okay, that's fairly straightforward. Good, that one was easy. So again, here, see, this is a typical example where where you really need this base sequence. Okay, you need to say, what the heck are you going to ask for here? I mean, so here, you really need to have said, I want, because I mean, I could want the base pair sequence for this. How do I know that I want the amino acid sequence?
But you give it a certain type of a. So I'm confused where there's an ambiguity.
Hold on 1 second. This is the VCF spec.
It's kind of ugly, but it seems to get the job done, and it does seem popular.
Well, okay. So, and maybe there's good, tools for handling sort of this question of, I want position 100,000, stack up all the indels before me, so to speak. But okay, back to myoglobin and what it actually is. In other words, first of all, I assume that is human myoglobin.
That's right.
Okay. So then the correct thing for this to return for biosequence myoglobin, I don't know what its correct thing to return is. I mean, I think you have to say, it might have a default, but, I think you'd have to say biosequence, and this is, again, this actually makes the case for the thing, so here we would say DNA and then entity. I don't know. I think I think these should be packaged somehow. I'm not quite sure how, whether it's like that or a pair or something.
But I feel like this concrete example is not so much different of molecule of chemical.
Yes, it is. Because a molecule consists of a bunch of atoms and bonds. That is the only thing it can consist of. Whereas here, this protein, well, okay. You're saying this protein is an amino acid sequence, so don't ask about the DNA. Use biosequence convert to get to the DNA.
Exactly. Because like you could argue, if you argue your way, then I would argue the chemical is not a molecule, but it is a many body system of infinitely many.
Yeah, right. Okay, so here, this would be biosequence of, and this would then be a peptide, and then it would have a sequence, whatever that sequence is, whatever it is. Fair enough.
If you start with a protein.
Sorry, what?
If you start with a protein entity, if you start with another entity, not. Like your left-hand side is just protein entity, not just any entity.
Yeah, right. Right, and if the entity is a gene, then this right-hand side encodes the base here is human genome DNA, and then the diffs in quotes give that particular location for that particular gene on the human genome. I think that's right. Aye. And we'll do that for the whole of my DNA. Why is it called sequence molecule? I think it should be called biosequence molecule if it's called anything. Why isn't it just molecule of that thing? Why isn't it molecular biosequence?
So I'm stealing this functionality from the chemistry folks, so Jason would have to weigh in here.
Let's see. So sequence molecule is an internal. I mean, at the moment, it's in the chemistry context.
Well, why isn't it just this? Molecular biosequence gives a molecule.
Yeah.
It's like, okay, that was simple.
Yeah. So I mean, internally, I will ask for the sequence and then feed it to the function sequence molecule that we've.
I don't care what you do internally. You can feed it to your pet function. But I mean, from the point of view of the user, this would then return a molecule. This is then converting it to molecule representation. Now, a more interesting question is biosequence of molecule, the round trip.
Right.
Yeah, let's do this one later.
Yeah, because wouldn't it actually have to be a sequence of molecules? Not, I mean, you'd have to have a list.
Because the whole magic would be to make the cuts right.
Ah.
Right. It would
right,
be
and I mean,
protein.
So we could cheat a little bit in that, and when we make a molecule from a biosequence, we could add meta information.
Tagging. Yeah. Yeah.
So right. I mean, just make it one of the properties in the meta information, storing the sequence, so that we don't have to recognize the sequence.
Yes, we could do random instance. I don't see a problem with that. That is a grotesque return value, in my view. I mean, I've never liked that. I mean, I invented it, but and I've used it a gazillion times. Well, not a gazillion because I haven't done
I
that.
don't like it either, but we followed.
Well, it's better. You know, I've used it on my own genome, for example, and I have to say it's not totally terrible. I mean, it's, you know, if I want to find where do I have SNPs, you know, I got this big gene, it's a megabase long, and I want to find my SNPs there, I can at least see very quickly where do I have variants.
But the inner list head I don't like.
Well, I understand. It's quite convenient, though. I mean, in my experience using it, it is not completely grotesque. I don't know. I have not come up with a better way to do it.
You don't think that just a rule in the inner one would read easier?
Yeah, that might be easier. That might be better. Either way, though, it's still a weird thing to break up what is ultimately just a string, put it in a list, and have weird annotations. I mean, it reminds me of what we're doing with things like text structure. What is the point here?
So for dense alignments, this is something that Trevor's used, where you have kind of each sequence is maintained, and then dashes are added, and so you have the original tracks and can work with the original tracks for further analysis.
I didn't understand that at all. But actually, I have another question. In this business about an acceptance threshold and so on, clearly, the matches that you've got to be able to do for the whatever the find biosequence thing is, okay, has got to include ends in the thing being found, and the thing, both the big and the small, could contain ends.
Yes.
So
yes.
So this is a weird case of pattern matching, because in ordinary pattern matching, it's like match this pattern which has blanks in it to this thing which is literals. Whereas what you're doing here is you're matching patterns to patterns. So in other words, if I, for example, have NNNNN, and I'm matching NN to NNNNN, then clearly that matches in many different places. Right? And it happily says it's rather degenerate, but it's going to say there are matches. It's NN with lots of alignments. But that's okay. I mean, I'm just pointing out something that should work, right? You should, and I'm not confused, right? I mean, you should be able to have both unknown, it could be anything, in both the big, even the reference genome has ends in it, I believe.
That's right. Yep.
So fair enough. Okay. All right. Similarity scoring. Great. Wow. Wow. Okay. That's we can, don't tell me, we can do string replace and it'll do a CRISPR for us.
Something to like.
If you like this, you must accept it against Idai.
I can imagine that I doesn't like this. I mean, and I also wonder about a rule like that. Because I worry that there may be some biology-specific transformations that are and are not allowed. I mean, that is a nice shorthand, but
I'm not a big fan of it. So I understand its usefulness, but I'm kind of, from a language point of view, not the biggest.
Well, you think it should be biosequence replace, biosequence reverse, etc.
Yeah.
I think that's a safer thing to do. Significantly safer. It can be cognitively similar to the string operations, but I mean, and by the time you got freaking circular DNAs and things, that's complicated. We don't have circular strings, and also we have a lot of options to string replace, which probably don't apply here.
And just yeah, this is cool. Just as an example, that this would come in a lot with is restriction enzymes. So often you want to know where restriction enzymes cut, and those will have certain particular motifs that they'll cut, which would then probably be like ATGN G, for example, and the N actually doesn't matter in that cut.
I see. Right. So it's got to be biosequence, replace, etc. I mean, just think like ignore case. What does that do for a biosequence? Who knows? It just doesn't make any sense. So make them biosequence, whatever things, I think. Now, what is this? Biosequence string processing. No, I don't like that. It's just, let's not think of them as a string.
That is just basically to not have too many own ones, a cheap way. It follows basically matrix function. Like we don't have all our 300 special functions repeated for matrices, but we cannot use them because they thread over lists. So that's why we have matrix function.
Yeah.
That's
this
basically
is
the
different,
equivalent.
but this is different because I mean, the string operations that you might want to do, oh gosh, string take.
They're exactly all that you might want to take.
Well, if I was doing it, I wouldn't have.
You can of course map at position 2, but that's ugly.
Right. So a couple of comments on our live stream. One is visualizer. Wouldn't a visualizer be better than this? Wouldn't a blob effectively that is a visualizer for this be better? And I agree. A blob, maybe with sliders and things that you could go and look through the sequence or zoom in and things. And I agree that that would be a nice thing to provide. Suggesting that there's a sequence alignment object or something that comes out.
That would be nice.
And we could actually, because sequence alignment is a useful function for things in general, used for all kinds. I've used it for all kinds of things that have nothing biological about them. We could imagine a biosequence alignment, which is specifically for biosequences and which has special features like coming out with a blob. I think that would be useful. Now, okay, we're going to need to talk some more about the string stuff. This is kind of grotesque. I mean, I feel like if you just want to do string operations, extract the extract the sequence string, do the operations, and if you want to, stuff it back in again. Because I predict that there'll be a few operations that really make sense, and a lot of others that really don't. And I mean, the question about, I don't think we should really distinguish complementation from reversal, for example, and having one be string reverse. We don't have a string complement because we don't say, B goes to Y, C goes to X type thing, or something crazy like that, right? So complement is very much a biological thing. Right. Reversal, and again, there may be, and reversal, I don't even know what reversal, okay. Well, someone on a live stream just suggested the exact same thing I suggested about transforming to string. SWSH is wondering if in the distant future there'll be a high-level language for expressing genes and their interactions instead of, as they put it, this obscure base code programming for life. Yeah, many of us have been wondering about that for a long time. Actually, a question for our biofolk here, just out of curiosity. Has anybody made progress on that? Of something higher level for expressing what's going on?
I don't think so.
Fair enough. It's assembler language.
Like it's just layers and layers of through encode of like nucleosomes and methylation and all of the things, and so it's just a big dictionary that's getting bigger and bigger without proper rules.
Right, and we don't know the programming primitives for life, really. I mean, we basically don't know beyond very straightforward things. I mean, we don't know, the analog of the functional programming primitives. We barely know. I mean, that is, what is the higher level primitives of, this restriction enzyme acts on this thing and cuts and splices and does this and that. I guess we know some of those operations. But is there a higher level notion of, these are the functional programming primitives that represent, interesting things that happen in living systems?
The problem is there's no guaranteed hierarchy or modularity to anything too, because it's just what evolution comes up with. And we try to put abstractions on it after the fact, but there's going to be plenty of rule-breaking things that don't fit nicely into a box like that.
Fair enough. Even as probabilistic kind of programming concept. I mean, this upregulates that or something. I don't know whether that's but that's not really a programming. Yeah. Okay, fair enough. Oh boy. All right, we need to discuss this some more. I don't know what the right thing to do is here. Okay, I think we're running out of juice.
Let us rework the segmenting.
Okay. This looks fairly straightforward. This one I think is going to be fairly straightforward. He says famous last words. I mean, transcriptions and translations and complementings and so on seem fairly straightforward. I would think that in addition to this, you might want an accessor here that says reverse complement string and so on.
Yes. Yeah. They don't cost anything.
Indeed.
What's the plan to approach things like secondary structure and that kind of thing? And
that's the first thing in this kind of broader functional picture work, extensions.
Okay.
So
got it.
What would you like it to be? I mean, and that is you're trying to say this RNA folded in this way or something. Is that what, in addition to saying this is its sequence?
Yeah, exactly. And that applies even to protein folding and quaternary structure and all that good stuff is classical and ripe for representations here.
Well, so I mean, the way we do that in molecule is we have this atom coordinates concept.
Yeah, so similar to like you can convert a sequence to a molecule, you'll be able to convert secondary sequence to structure in terms of atomic position and that sort of thing. But you want higher level, you want to be able to say like this is an alpha helix, this is a data sheet. You want to be able to say this is what like an RNA fold looks like.
Right.
In terms of Watson-Crick pairing instead of actual atomic positions and all that.
Yeah. You have an MD simulation, these are the water atoms, this is like how your protein is flopping.
Yeah, I mean there's things in we'll do like folding of RNA is just a function that takes RNA in and spits out a bunch of secondary structure.
Right.
It's a super useful function that gets used.
Which can actually be done for RNA as opposed to
right, exactly. Exactly. It's not nearly as hard a problem when you're just dealing with Watson-Crick pairing instead of tertiary interactions.
But so in our case, then, what I would imagine is biosequence. I don't understand why biosequence, like molecule, I mean, molecule has a logical graph theoretic structure independent of its spatial, these are the positions of the atoms structure. And I don't understand why we wouldn't do the exact same thing for biosequence. It has a logical chain structure, but then it could have coordinate positions that are annotated into it. Is that an obviously wrong thing to do? And then you can ask questions. I mean, these questions which we don't get to ask on a molecule. Okay, so let me ask this question. Okay, I've got a molecule. I want to know, I got this big molecule, and I want to know, will this random molecular proboscis fit in my molecule anywhere? How can I answer that with our molecule setup? In other words, it is a computational geometry type question. Is there a hole in my, if I've got some random...
Docking is a full thing.
Yeah, this is docking.
Yeah.
But you do want, before you get straight to that, you do want some kind of modular higher level representation of secondary structure before getting down to atomic position, because it's still referred to in, like you'll have things that you want to do homology for predicting, machine learning to say, this hidden Markov model says it's probably an alpha helix here versus a beta sheet.
I see.
Those are kind of classical comp bio problems that you're going to want to tackle.
But so that's similar to the annotation rules thing, where you say this is methylated in this area. This is an alpha helix in this area. Isn't it?
Yeah, you could do it that way. I mean, that's basically how, when we do folded RNA DNA, there's kind of a secondary list of the bond pairs of what goes to what. in order to be able to draw a structure and say this is what it looks like in 2D.
Okay, fair enough. So I mean, you're saying we should be able to use annotation rules to annotate, like I don't know whether there's alpha helices and beta sheets and things, and I'm sure there's. I mean, I've even seen these sort of periodic tables of weird repeated structures that exist in proteins. So you could say, and I'm going to annotate this as a, head and shoulders configuration inside my protein or something. Isn't that right? I mean, that I might want to annotate it with other sort of facts. You're saying that are less specific than it's this atom position.
Annotation could, in theory, be used for anything, right? But whether you elevate it to a higher level of, so there is a construct for it, might be worthy of consideration because it is.
I agree, but I think with annotations, we have this convention that you can have, there are standardized keys for an annotation given as an association. And I think that's a possible approach to that, which is both extensible, as people choose to do different kinds of tagging and can handle known kinds of tagging in special ways.
Yeah, good work.
There's a question here about missing inside these things, and yes, we should deal with that. We should deal with having missing turn into ends. In other words, the symbolic missing should turn into an N, and in certain cases, we might want, as a matter of fact, we might want to, just like we say sequence string, we might want a sequence list as well that has things like that in it, possibly.
That makes sense.
Brian is asking, how does similarity scoring treat insertions, deletions, substitutions? That's a whole elaborate story. SWSH is commenting about the hierarchical nature of it's all physics eventually. The terrible thing in my physics project, which I'll be starting to talk about hopefully in a few weeks, is you talk about the need for higher level programming languages for things. This is a extreme version of that where it's just built out of unbelievably abstract things, and the question of how you think about it in terms of something that one can seriously discuss, so to speak. Like biology has never solved that problem. I hope we'll be able to solve that problem for fundamental physics, but we'll see. Trito was saying figuring out the shape of a protein from a amino acid sequence is not quite solved. I think that would be a fair statement. Although I don't know whether, as Trevor probably knows Mohammed Al-Qurashi, who is former employee of ours. Have you guys have interacted with Mohammed in these conversations? Yes.
Yes, I've had the pleasure to talk with Mohammed about this. I'll also note that Trevor has dropped off the call. Unfortunately, he had a commitment.
Okay. Well, anyway, so Mohammed claims that he has really made great progress using machine learning and other things in this protein structure thing, which is a long-running, difficult problem. But okay, so we got lots of stuff here oh boy, lots and lots of stuff to talk about. Okay.
So we rework it and then we can get together in a couple of weeks.
Sounds good. I think we didn't completely solve. So we got one biosequence thing, and we've probably got this find biosequence and biosequence.
And we still, I feel like, have to deal with the alternative.
Yes.
But there's a much broader issue outside of biosequence, yeah.
Right. Yeah, right. I mean, it's the categorical alternative, so to speak.
Might be even of relevance for a return value for certain entity value things where we don't know quite what it is, but we have narrowed it down to a couple of alternatives.
Well, I mean, the obvious name that we could use, no, I don't know. There's either. It's a bit two-sided, though. Anyway.
Yes, it's a little bit too. I agree.
All right. Well, very interesting stuff. This is great. I mean, this is a wonderful fascinating thing.
So
And,
another 2-hour meeting.
Yeah, I think we can assume that
Okay.
it will be that. I mean, it's a big area, right? I mean,
Yeah.
how long did it take us to do chemistry? How many hours of meetings did it take us to do the design of chemistry? I'm
10?
guessing.
Jason.
I would say 10. Not more.
I think it was more than 20. I think it's more like 20. Which is modest, honestly. It's okay. These are big areas. It's all right. Well, look forward to it. Thanks, everybody. Thanks.
Sounds great. Thank you.
Yep.
Outside folk. Thanks a lot, everybody.
Thanks.
See you all
Bye.
later.